Good day, ladies and gentlemen. I am Dr. Kingsley Abhulimen from the Department of Chemical Engineering, University of Lagos. I'm also a Director of Technology and Innovation at the University Technology System Limited. I'm going to be speaking on a project or a research we've been doing over the last 6-7 months, and which is a joint venture between the university and St. Texas. And basically, it's on the COVID-19 pandemic. Basically, we have come up with a therapeutic method for immunotherapy vaccine treatment of novel coronavirus epidemiology in the human system. Basically, I'm not talking about a vaccine that we can produce outside the human body. I'm speaking about a vaccine that the immune system itself will produce on our body, and it will help us build the antibodies to fight this virus. And basically, I know there have been a lot of race to get a vaccine, and there have not been any successful clinical phase 3 trial. But all of this vaccine outside the human body will fail, as our research have clearly shown, because it's like modeling the solar system of the universe, the Milky Way, Andromeda, and all these other galaxies. And think about the body system as that number of genes, about 12 trillion genes in the human DNA system. How do you want to manage a vaccine that has been produced outside the human body to be able to understand how the whole 12 trillion genes work? So we have come up with a method for clinical immunotherapy using vaccine treatment of novel coronavirus. That's the research we've been working on. So I'm going to introduce you to a prototype we have developed, and this is the prototype, and this is the cardiovascular system of the human body, showing all the blood vessels. And we're going to be understanding how this machine produces this human analysis for the purpose of doing these simulations. Basically, I also manage the Energy and Environmental Research Center of the University of Lagos. This paper presents model algorithms and clinical manifestations of clinical simulations of viral antibodies mapping in the human DNA system related to COVID-19 epidemic pathology. The model algorithms are clinical expectation based on Bayesian probability derived from clinical data obtained from the Lagos Hospital in Lagos State, Nigeria, of some COVID patients. We also took data from the Wuhan, China report that was published in the literature in sometime in March 2020. Well, why we're looking at the Bayesian model is because we want to be able to say, okay, these are the clinical expectations we have, given the set of symptoms which we call the operating conditions, to give a kind of probability of whether somebody can contract this virus, or whether somebody has it, or whether somebody is going to live, or whether somebody is going to die of the virus. Now, the model algorithms integrate randomized viral infection manifestation from published clinical data to simulate human genomics sequence and bioinformatic evolution related to infected patients in a MATLAB software environment. So we have to understand the whole genomics. And the way we have modeled this like what we have in the Milky Way, where we have the solar system and we have the earth realms, that's the material universe, and the solar system. And the solar system is controlled by water, and the material realm is controlled by matter and what we have. So what we have basically is a fit of clinical expectations derived from the infected immune antibodies index for 4 patients' cases from the clinical data we have derived. And the infection to the immune mapping vector of antibodies evolution is based on preclinical data, was further developed in the MATLAB software environment where patient 0, the primary carrier of the COVID-19, exhibited the highest degree state of 47%, followed by patient 0, patient 1 exhibited 45%, patient 2 exhibited 6%, patient 3 exhibited 3%. So with our software, we'll be able to determine the clinical expectation, whether somebody will get a COVID or whether somebody has it or whether somebody is going to die of it. So we have developed a system that allows us to do diagnosis. And the probability model is taken from 0 to 1. And it's represented by a Fusey class belief system, which we have also developed to help us understand whether our data is correct by subjecting it to some belief system, which we have designed also. So that's just a summary of the research, and we're going to be presenting the results in subsequent slides. And basically, the COVID-19 biomarker in the human immune pathology targets single nucleotide polymorphism of the infected patient physiology, observe true clinical disease symptoms, manifestations. The only way we can determine whether somebody has a COVID beyond just testing is that the patient begins to exhibit some symptoms that are akin to COVID-19 epidemiology. According to a report that was published, a novel respiratory virus originated from Wuhan, China, has spread to more than 100 countries combined, located in Asia, Europe, North America, Africa, and Middle East. More than 3 million cases have been reported alone in the United States, and up to 20 million cases have been reported worldwide, and there are about more than 180,000 deaths as we speak. And this obviously is the World Bank and the World Health Organization has probably declared this as one of the worst pandemic we've had. Now, the economic impact of the coronavirus epidemiology to countries, especially the United States, Europe, and the Asian countries, translate into trillions of US dollars in lost revenue, and also $3 trillion in monies that were actually spent in trying to curtail the virus. So this represents an enormous economic downturn. Not talking about the loss of jobs and all of that due to the COVID-19 pandemic. Mortality rate has increased in some clinical laboratories, and it has caught the attention of governments and world bodies to try and come up with a solution. I know there's a race to get a vaccine, but that is not going to work. Basically, we're going to look at some of the issues that are related to that. Now, the Australian hospitalization for this is about 100,000 rates. Basically, the virus is effective in transmitting through communities, through airborne or physical contact, with experts confirming doubling every 6 days according to published model. Much remains unknown about the virus. However, many people have very mild or asymptomatic infections. The economic impact of the virus to the states and translates directly and indirectly into trillions and trillions of dollars. Now, one of the ways we have modeled the human DNA system, we had to take a simple model, our solar system, that's the Milky Way, and come up with, how does the DNA of the human being looks like. So model the human DNA system as a constellation of solar system of suns and stars in an ICU 1101, Godzilla Triangulum, and Andromeda galaxy, infused in a human Milky Way Earth realms consisting of 144 trillion stars, 100 million suns that we will say constitute what we call the gene of the DNA system. That is quite complex. And we want to be able to model, say this is a hypothetical model. And you're thinking of making a vaccine that will try and check up all how these things. There's no human that can understand this, how it works. But we're going to try and provide some truth in this thing. So we want to say this, that the human DNA system carries the generic of like galaxies-like system. And when we understand this, we can be able to understand how the immune system provides the vaccine for a lot of the diseases that we have in our body. So we must be able to understand how the immune system reacts before we can be able to defeat this virus. And that is what our research have come up with and trying to propose some solutions to that. Now, the DNA genomic sequence organizes antibodies, bioinformatics evolution into clinical disorders and reorders of the existing DNA strain. That is very unique. That means that it takes your original DNA system, it takes the virus and tries to understand what this virus is and maybe provide antibodies that can check the virus. And basically, the COVID-19 is not a disease. It's just a single RNA trying to invade into your whole DNA structure. So think about one single cell trying to invade 12 trillion combinations of genes. That is awesome. And the virus begins to multiply as it sees the DNA trying to attack. And it's like the immune system is trying to build a defense for the virus. And so it's like if we try to build a 12-foot wall and the virus builds a 14-foot, then it's going to be something like that's happening between the DNA and the the virus. And eventually whoever takes over kills. But I'll tell you the truth, it is not the virus that destroys you, it's your immune system. The immune system overpowers the virus in a way that it can destroy your liver, it can choke your skin and cause feverish things. So we have to understand how this works. The COVID-19 virus RNA penetrate the donor DNA genomics and bioinformatics. We're trying to code of the donor and mutates the donor to propagate itself to gain dominance of its host in the complex DNA structure. And the DNA genomic sequence produces antibodies to try and check this infection. And basically, we are going to look at the major theory gaps in genomic organization and bioinformatics. Major theory gaps in knowledge of origin, epidemiology, duration of human transmission, and clinical prognosis of COVID-19 viral infection pandemic has necessitated further research in the finding the appropriate clinical technology applications to simulate antibodies to limit virus population, propagation, transmission, and epidemiology on the DNA gene framework. The subject of genomic organization and bioinformatics, evolution of the human DNA, and the virus RNA falls under a subclass of DNA genomics, protein, bioinformatics science class. Coronavirus are envelope segmented positive sense RNA viruses belonging to the family Coronaviridae, and other nidovirals are broadly distributed in the human and other mammals. Coronavirus is a single RNA protein surrounded by lipids consisting of fatty acids. So coronavirus is not a disease. Coronavirus is just a single protein surrounded by fatty acids. And so you cannot be treating the coronavirus as a disease. Start treating it as if it is one single DNA strand trying to propagate itself in the complex human DNA system.